Today, the Royal Tyrrell Museum and its cooperating society are proud to present Dr. Kevin Seymour. Kevin is the Assistant Curator of Vertebrate Paleontology and Collections Manager at the Royal Ontario Museum in Toronto. But Kevin also carries on a secret double life as an orchestral musician on evenings and weekends. Kevin was born and raised in Ottawa. He obtained his bachelor's degree in biology and geology at Queen's University in Kingston, Ontario. He then moved to Toronto to pursue his master's degree in geology at the University of Toronto, where he studied fossil cats preserved in tar pits from Peru. After completing his master's degree, Kevin was hired by the ROM, and then after a few years working at the ROM, Kevin decided to pursue his PhD in zoology at the University of Toronto, where he studied the morphology and evolution of South American small cats. Kevin's research interests revolve around the study of quaternary faunas of eastern North America, especially in Ontario, and the evolution of North and South American cats. Over the years, Kevin has conducted field research in Canada, the United States, Mexico, and recently South America. Today, Kevin will discuss his involvement in the study of an exceptionally well-preserved 52-million-year-old fossil bat in a talk entitled What the Fossil Record Tells Us About the Evolution of Flight and Echolocation in Bats. So without further delay, I present you Dr. Kevin Seymour.
Does that mean you can hear me, or should I? Is that fine? Yes, okay. So you may wonder, for someone who normally works on cats, what I'm doing with bats. But it's only one letter change, so we'll go with that. Okay, so were they going to turn down the lights maybe a little? He's gone to look for the light switch. There he goes. Okay. I understand you had a speaker on bats a few weeks ago, so you know a little bit more about Alberta bats now. In terms of the world, bats are one of the most diverse and largest radiations of mammals. They represent approximately 1/5 of the world's mammals, or about 1,000 species, spread across 17 or 18 or 19 families, depending on which expert you listen to. They're diverse in terms of what they eat. Many of them, the ones we have here in Canada, are insect eaters, but some tropical bats eat fruit or nectar, and there's even a couple of species here that actually catch fish. So they are relatively diverse in their adaptations, but they, in common, have two things that are unique to bats, and that is their ability to fly and their ability to echolocate. And echolocation is the ability to use sound to locate prey. Now, even though the fossil record of bats extends over 50 million years, the early evolution of this group of animals is really poorly understood. Those two key innovations, echolocation and flight, how did they evolve in this group is not known. And that's because these fossil bats, these ones from the Eocene, around 50 million years old, basically are essentially living bats. There's hardly any difference between them. And these, they basically look like bats. And studies have shown that these bats could echolocate. So in terms of where did the bat come from, these fossil bats don't tell us anything. They're perfectly good flyers and perfectly good echolocators. So how did these things evolve? We didn't know until now. This is also, I was going to mention that these early bats have stomach contents, the ones that are preserved from Germany, and they have insect parts in their stomachs, and we can see which kinds of insects they were eating, and some of them were eating beetles, and some of them were eating moths. So even in the early Eocene, the late Eocene, these animals already had specialization in terms of their kinds of prey that they were catching, but they're all flying prey, flying beetles or flying moths. So up until now, the fossil record really didn't tell us anything about how bats evolved until 2008. And that's when we published this article on this new fossil bat. Now, apparently, the press really thought that people love bats, because when we published this, within the first day, there was this feeding frenzy of press. It was reported in 158 newspapers worldwide. That was just the first day, and then I lost track. And so either they think people really like bats, or else it was a slow media day. I'm not sure which, but they came out with some really bad titles, like Holy Missing Link. And my favorite was from the New York Times. Primitive bats took to the wing, but they didn't have that ping. So people are very creative when it comes to newspaper articles. What they didn't really emphasize was the Canadian connection to this. So that's one of the things I'm going to tell you about today. And that is because the holotype, or the prime name-bearing specimen for that, is a Royal Ontario Museum specimen, the one that you see here on the left. On the right is a second specimen, which was the first collected specimen, and it is in private hands still. And so it was not available by the rules of nomenclature to be named because it was in private hands and not in a public repository. But all of us have got casts of this particular specimen, and it's more nicely laid out on the slab. So I'll point out some features on it versus the other. So I'm going to use them interchangeably, but there really are two specimens. In terms of our gallery, we display the cast of the right-hand specimen because it's more pretty. But the left-hand specimen is the prime specimen because it has a beautifully complete skull, and I'll go into the details of that one. Now, why does the ROM have this specimen of a bat from Wyoming? It has to do with our new gallery that opened 3 years ago. And leading up to that gallery opening, we attended this Tucson Mineral and Fossil Show for several years in a row to purchase and obtain specimens for our new gallery. These are some pictures of us at the fossil show. This is my colleague Dave Rudkin here, and this is a nice big slab of trilobites which we did purchase for our new gallery. This is my colleague Janet Waddington, and this is a Cervalces skull. This is an Ice Age moose-like animal. It's a cast. We ended up buying that whole skeleton for our gallery. This is me besides a whatchamacallit. I don't know what it is. It's just something somebody carved and it looks funny, so I wanted a picture beside it because I don't know. It's just a piece of art, basically. And this is a marine reptile from China, which we did not purchase because it was illegal. So we had to be careful when attending this show. Some specimens were legal and some were not. But one of the things that's most commonly displayed at this fossil show are fossils from the Green River Formation, and that's the formation that this fossil bat came from. So I'm going to take a little sidestep here into the Green River Formation and tell you a little bit about it. It occurs, outcrops in 3 states in the US, Wyoming, Colorado, and Utah. So the white areas here are where the rock outcrops in those 3 states. The ROM collected this area in 1969. These are some pictures of the ROM Expedition. And primarily, almost exclusively, we collected fossil fishes from this location. So you can see the effort here. It's very nice, thinly laminated shales that you can split. You can see here a whole really long, big slab has been split and lifted up, and you can find complete fossil fishes on them. These are some of the fishes that we collected in 1969 that have since been prepared, and all of these are on display in our new gallery. The stratigraphy, or the layering of the rock here, gives us evidence that these fossil deposits were lake deposits, and the ROM collected here in the F1 deposits, and this new fossil bat is down here from the lowest level. And the fossil deposits show us mass mortality layers where thousands and thousands of fish are preserved in one layer. And by studying the rocks, people have concluded that they probably represent what we call a mass mortality layer due to an algal bloom. So in this area of these 3 states, there were a number of inland lakes, and at certain times of the year, there would be, because of the nutrient balance, there would be an algal bloom, and the algae would suck all the oxygen out of the water, and all the fish would die in that particular local lake. And because of the algal bloom and the changing of the chemistry of the water, by removing the oxygen, the water becomes slightly, let me think, is it more acidic? And then calcium carbonate precipitates out. So it's a beautiful system. you can kill off all the fish and cover them up with sediment at the same time. And because of that, this area is world famous for producing absolutely complete fossil fishes, every scale, every fin ray in place. And so they're very famous, and hence they're sold worldwide. This is Bonnie Finney, who collected the new fossil bat at the location where she collected it. In 2003, she's pointing to the layer where it's found. She's not pointing to the Coke 10 there. And she did 4 really amazing things that enabled this purchase to happen. Because I had been attending the Tucson show, and there are 7 dealers that have quarries, licensed quarries in the Green River Formation, I would visit all the dealers, occasionally buy a fish from them and talk to them. And because this cast of the first bat was already available, it had been collected, I went to all the dealers and said, if you find another bat, you call me, because we really want this bat. It's a really important thing, and we don't have access to the first specimen, but if you get a second specimen, call me. And that's exactly what she did. So she collected this in August, and first thing she did was call me. The second thing was she held onto the specimen for 5 months until I could raise some money to purchase it for the ROM. And the ROM has a trust account, basically, of a big an estate that was donated to our museum, and the interest from that estate can be used for purchase of specimens. And so this endowed money is available for purchases, and I had to apply to it to get the money. So she held on to it while I was getting the money and didn't sell it to anybody else. She also made an X-ray of the specimen before it was prepared to prove that it was all there. And she resisted preparing it. That had been another request of mine to all these dealers. If you find this thing, let us do the preparation. I was fairly convinced that the first specimen that was still in private hands hadn't been prepared properly, and some of the critical features were not visible. And so if another one was going to be found, I wanted it to be prepared properly in our lab. So she did all of those things, and hence we named this new fossil bat after her for her cooperation. This is what the bat looked like when it came to us. Not very incredible. If you squint, you might be able to see. So this is a piece of the wing. This is the head right here. Here's the forewing, you can see here. And here's the other forewing there. And then, so there's just the tips of the wing preserved. And she looked at that and thought, I think I see a little claw there. I think that's one of those bats. She X-rayed it to confirm that the whole skeleton was there. And on that basis, we purchased this specimen. Doesn't look like very much, but it looks much better when it's prepared. Now, the unsung hero in this story is Ian Morrison, our lab technician. He did a superb job in preparation of this fossil, which he called the bat out of hell, because it had an unusual preservation. In the middle of, if you think of this squashed bat on a layer of rock, in the middle of the bat there was an actual gap. So as he was preparing away, suddenly he would poke through the little gap in there. And that meant that you couldn't prepare it out completely, or the bones would just fall into this gap. But as they say on Star Trek, resistance is futile, and in the hands of this expert, he managed to do a fantastic job, even though he admitted it drove him a little batty. So this is what happens, is that some of it's prepared from one side of the slab, and some of it's prepared from the other because of this unusual preservation. It also means that here in the main body cavity, he didn't finish the prep, because if he had, the ribs would have fallen into space and just fallen off the specimen, because the gap is particularly well developed right here in the body cavity. So the all-important skull and the sternum are prepared from the other side of the slab, and the main body is prepared from this side of the slab. Because of that, it's not a very good display specimen. You can't show it on both sides at once, and so we put the cast of the other specimen into our gallery. So let's look at this specimen a little more carefully, and I'll go through some of the features why we think this is such an unusual thing and why it helps us tell about the early evolution of bats. Okay, so the first thing is the limb proportions. If you look at modern bats, they have the hallmark of very short hind legs. Both the femur and the tibia here are quite short, and because they're flying, The humerus is relatively short, but the radius and ulna are really long. So that's this really long middle part of the arm is so that they can fly, and the short hind legs is because they roost by hanging from their hind legs. Now this bat is completely different. It has quite long hind limbs still, and a relatively short forelimb. So when we graph that, to put it in context of other bats, this is what we get, this graph. So this axis here is a proportion of parts of the front limb, and bats have fairly long ends of their wings, so they plot out here on the far distance. This axis compares the forelimb to the hind limb, and because of the way the ratio is made, those that have really short hind limbs fall up at the top of the graph here, so up high. So here is where all the modern bats plot out. The little black dots that you see are fossil bats from the Eocene, and the yellow dots are other bats from the Eocene, the same location as this new fossil bat. So even those all fall into the cluster of data, as I mentioned, that are bats, and that's why the fossil bats up until now didn't tell us anything new. They just look like bats. Here are non-flying mammals, so these are typical terrestrial mammals. These ones here are terrestrial mammals that have elongated forelimbs, including the tree sloths and the primates called gibbons that have very long forelimbs. So they are ones that are either swinging or hanging in trees. And here is where the new fossil bat is. So its limb proportions are unlike any living mammal, either terrestrial or flying. So let's look at the proportions of the wing next. When you come to analyzing recent bats, many people have done studies of bats looking at correlating the proportion of the wings and the shape of the wing and how that tells us how they fly and what is their feeding strategy. So this has been, there's many studies on feeding strategies of bat correlating with structure of wing. So we can now look at this fossil bat and have a guess about how it was foraging. What we do for that, or there's several things that are calculated. One of the things we do is to compare this thing, this area here, which is called the hand wing. These are all the finger bones out here. So this part is called the hand wing versus this main part here, which is what we call the arm wing. So it's suspended underneath the arm bones, and these are under the finger bones. So what we're going to do is just compare the size of these two pieces of membrane. Now, we don't actually have a membrane imprint for this bat or any of the fossil bats, but what we can do is measure the actual bones, and this bone, the two finger bones from digit 3 and digit 5 are the most useful. If this bone is really long, then we get a fairly large area for the arm wing, and if this bone is fairly long, then we get a fairly large area for the hand wing, and vice versa. So just by looking at the measurements of these two digits, we can get a fairly good handle on the shape of the wing and therefore make a guess as to the foraging strategy of these bats. Now, living bats, this is a thing called aspect ratio when you contrast this area versus this area of the wing. Living bats, when you do this measurement, spread all the way from here to here in this aspect ratio. And basically, these ones up at the top are ones that have very long tips of the wings, and those tend to be the fast flyers. They tend to be foraging above trees and catching insects on the wing. And the ones at the bottom of this area are the ones that have short tips and fairly broad base or arm wing. And those are bats that are slow flyers and quite maneuverable, and they tend to be foraging within a closed canopy or in areas where they're sort of maneuvering slowly to catch their food. Here is where the Eocene bats plot that were known up until recently. These are the ones from Wyoming and Germany. And they already fall into those two categories. There are some with these long, narrow wings that were probably snagging insects in the air above the canopy. And there are some that have the shorter, broader wings that are more maneuverable and probably foraging in a different way within a closed canopy. So the fossil bats known today already cover almost the full range of what extant bats do. But our new fossil bat falls down here. So it falls off anything that living bats are doing. So it's another piece of evidence to say that it was doing something different, and it is closer to being a missing link than what we've had up to this time. We can also plot this. So here's the same idea, measuring basically the areas of the two parts of the wing. And here's where our new fossil bat, those are the two specimens, they fall here, again, off the range of all the living bats today. What I'd like to talk a little bit about are the bats that are closest to this fossil bat, and these are the rhinopoma bats, these two green dots here. So these black ones are all fossils. So the living bat that's closest are these green ones. Now, these are called the mouse-tailed bats, and they have a fairly small distal part of the wing here, or the hand wing. And they also show a very peculiar kind of flight that no other living bat shows. And they have this ability, or this habit, of flapping and then gliding a bit, and then flapping and then gliding a bit. And this suggests to us that since this is the living analog that's closest to our fossil bat, this may be the way our new bat flew. But it also suggests that gliding may have been the ancestral state of bats. So this is, you can see or imagine a gliding animal first, and then a little bit of powered flight mixed with gliding, and then ultimately full powered flight. So let's go through this fossil bat and look at some of the more details, the details of its bones, to see why we think that this is what we think it is. So the first thing is to look at the hand, which we call the manus, and it has a long set of digits, just like all living bats, but most interestingly, it has claws on all 5 digits. So living bats have at most 2 claws, digit 1 and digit 2, but no living bat has claws on all the rest of its wing bones. So this is very unusual. And at a glance, right away, the first time we saw that, we knew that this was more primitive than any living bat, because no living bats have those claws. Second thing is the rib cage. Bats, because of the way they fly, the flight muscles and breathing and echolocation are all linked together. And in so doing, they have this short, squat rib cage, as does this fossil bat. Now, yes, it is squashed flat on a rock, but nevertheless, it still shows a typical bat rib cage structure. The next thing is the scapula, or the shoulder blade. There's an extra facet on the shoulder blade of all the living bats, and this bat also shows that facet. And on the clavicle, which is in us the collarbone here, it connects the wing to the shoulder, and it is robust in bats as a brace for that flying mechanism. And our new fossil bat, Onychonicterus, has that robust clavicle. And if you flip it over on the other side and look at the sternum, which is in us, the breastbones here, the sternum on Onychonicterus has a keel. This is a very similar concept to the keel in birds. It reflects the muscle attachments on either side that are part of the flight mechanism. So that, in concert, all of those features tell us that this is a flying bat. One of the really interesting features about this fossil bat is that it has a bone called the calcar. It is a funny little bone that comes out of the ankle. On a living bat here, you can just see it at the edge, and it helps support the tail membrane of the living bats. Now, this is the first time a calcar has been seen in any fossil bat, and because it wasn't preserved in any of those previously known Eocene bats, it was assumed to have evolved later in the evolutionary scheme of things. Present-day bats use this tail membrane as a net to catch their food. So they'll fly, they can echolocate, they don't always just grab it in their mouth. They sometimes snag the moth or flying beetle in their membrane and then eat it out of the membrane. The reason it's probably not preserved is when you look at it carefully, it's actually made of cartilage. It's not good ossified bone. It's all got a bubbly texture of cartilage. And so This bat is so well preserved that even that little cartilaginous bone is preserved. So, as I mentioned, it strengthens this membrane, not found in any other bat so far. And so people thought that this calcar bone evolved late in the evolution of bats in concert with that insect net catching ability of the tail membrane. Now we have it in the most primitive bat known in the world, and we now know that the calcar evolved before echolocation. I'm going to discuss that in a few minutes. So therefore, that this bone was not a part of the insect catching mechanism, most likely, but it was probably used to support that tail membrane, and therefore it has something to say with early flight. And so that was a really neat byproduct of this bat, is that it had this funny little ankle bone. Because of all this, because of all the morphology that showed us that it could fly and that it did not have echolocation, which I'll get to in a few minutes, we proposed in 2008 that this thing could fly but not echolocate. And that, in terms of the theory of how bats evolved, was a bit controversial. Many people had proposed that bats learned first to echolocate, perhaps sitting on a branch and echolocating, finding their prey, going out and catching it, and coming back and sitting on a branch. And that later they learned to fly. But this bat now shows us that they learned to fly first and echolocate second. Now, 2 years later, in 2010, a second group of people challenged our hypothesis and said, we think this bat could echolocate. And so what we had to do was re-examine this feature. And so now I'll go through the various features that we use to understand how this bat could echolocate. Now, I'm going to use some technical terms, but I'll try not to. I'll explain to you them in English. So, remembering that when you want to infer a fossil behavior of an animal, we don't have the actual behavior fossilized, so we have to make an inference. And what we do is look for morphological features, in other words, features on the bones that can tell us if living bats have them and this bat has it, then we make this inference that it would behave in the same way. So remember that this is only an inference because we don't have the behavior of these things. Okay, so there were 4 features that were proposed. We proposed the first 3, and Veselka et al. Proposed the 4th one. Now, these look like a bunch of gobbledygook, so I'll explain what they are. But they're all those features are based here in the base of the underneath of the part of the skull. So we're going to do some close-ups now and look at those features. So here's a bat skull that's flipped upside down. And I've color-coded the 3 regions that are of interest. The first one is the cochlea, which is basically the ear region, the inner ear region. The second one is the malleus, which is one of the ear ossicles in the ear of all mammals. And it's used to help transfer the sound into the ear, cochlea, ear region. And the third one and the 4th character, both those characters are involved with this bone, this blue, what I've colored blue here, called the stylohyoid. It's a hyoid bone, and mammals have hyoid bones that connect the tongue to the ear. And it's used also in terms of, in function, it's used to help create those sounds from echolocation. So this is functionally used for making the sounds. This is for receiving it and transmitting the sound into the ear. So this is the complex that's of interest to understanding whether something could echolocate, are the features located in these 3 bones. So we're going to look at each one of these in turn carefully. Now, in order to do that, because this is a squash bat on rock, we had to do X-rays, and this is where Jorg, my colleague in Germany, came into play. He has this setup where he can X-ray at many different angles. And ultimately, now, he can do CT scans on it, which I'll present in a moment as well. But originally, this was done with just a series of X-rays that were then assembled together. And so one of the things we want to see is that is the cochlea, the ear bone, enlarged as in all living echolocating bats? So we can get a scan of a skull like this and then actually measure very in good detail that first turn in the cochlea, which is where all the information gathering happens when they're receiving a signal. we need to compare that with some measure of size of bat so we can kind of take out the size. So what we do then with similar scans is measure this width across the base of the skull. So we're going to compare those two measurements to see if this region is enlarged in the fossil bat. So this is the kind of plot we get with all the living bats on it. The blue ones are the echolocating bats, and the orange ones are the non-echolocating bats. So there are, particularly the fruit bats, are those that don't use echolocation to find their food, and so they don't have a big cochlea. And this is where Onychonicterus, our new bat, falls, actually below the line for the fruit bats. So that says it has a small cochlea. It doesn't have the enlarged ear region of echolocating bats. The next feature is in the ear bone called the malleus, which is this one here. So we're going to zoom in. Again, this is an upside down skull. You can see the teeth of the bat here. And that's the region where here's the ear region in the hyoid bone. So we're going to zoom in on this. And there's a particular little process here on that bone that echolocating bats have. Now this, you can imagine, might be difficult to find in a fossil bat. When we first proposed this, it seemed to be a good correlate. But since that time, people who have been studying moles have found that some moles, those with the green arrows here, do not have this feature. But other moles do have, those marked in red here, do have this same feature. So in biology, if we can find an exclusive correlation, we can be confident in it. But in here, with these moles that are not echolocating, some have it and some don't. So we feel that it's not a secure inference to be saying that this is a useful feature for understanding echolocation. So it isn't necessarily an indication of echolocation because some moles have it and they don't echolocate. So in terms of our 4 features, we're crossing that one off the list now and saying we'll concentrate on the other 3. So we've already talked about the enlarged ear. The next 2 characters have to do with that throat bone, the stylohyoid bone. So they're both involved with the same bone. And they talk about an expanded end or an articulation with the tympanic. So we'll look at those a little more carefully now. So here's a scan of a bat from underneath. So you can see you're looking in its throat area. So here's its neck and here's its throat. There's the ear colored in yellow, or the tympanic bone of the cochlea. And then here's the chain of hyoid bones. And I've colored the stylohyoid in blue in this case so that you can see it. And for this slide and the next few, well, I'm going to be using the same colors, the yellow and the blue, so that you can see the comparison. So that's where in the body we're looking. If you didn't have this slide, the next slides wouldn't make any sense. So here are some details. Up here in the red box are what the fruit bats look like. They have the tympanic part of the ear and the throat bone, but it's not connected to the tympanic, and it doesn't have an expanded tip. So those are non-echolocating bats, and they don't have either of those features. Whereas here, the lower 4 are examples, these are CAT scans, by the way, of ear regions, and you can see that all of them, the stylohyoid bone is clasping around the tympanic, and it's got an expanded tip. And so this seems to be a pretty good correlation for if you are an echolocator, you have these features. But the question is, do some bats potentially have one and not the other? So are there bats? So what we did was go through all the world's bats, multiple species for every family, combing the basic morphology of these animals, looking for some bat that might have the expansion but not the connection, or the connection but not the expansion. So we're trying to understand this character. Do they always go hand in hand? And the answer is yes, they always go hand in hand. So that the fruit bats that don't echolocate have neither of these features, and all the echolocating bats of the world have both of these features. So we would rewrite our characters for understanding whether an animal was echolocating to really be just 2. An enlarged cochlea for receiving the signal, plus this double-barreled description of the stylohyoid bone, that it's expanded and connected to the tympanic, that those go hand in hand. So now let's look at our Onychonicterus bat and see what it has. So this is a scan of the underneath of the skull. I've colored the tympanic kind of a pale orange and the stylohyoid in a dark orange. Veselka et al. Claimed that this hyoid here was in the correct position to be clasping the tympanic, and therefore it might be an echolocator. And we say, well, look at the other ear. It's not in the same position there. You can see it also it has a tiny little knob on the end. We propose that that might be actually one of the other hyoid bones fused on there. That is yet to be determined. But that this is a squashed fossil, and those throat bones aren't like arm bones that are tied together with tight ligaments, that when this thing fossilizes, the thing gets squashed and crushed and things are a little bit out of place. So we feel that just because this is sort of more or less in the right place doesn't mean that it was an echolocator. So we actually did some further scans to show that it wasn't intimately connected. So this is just, we want to look at both of these stylohyoid bones. So here's some scans, the same coloring, showing that the tympanic is in one color and here's the stylohyoid bone. It also shows that it's broken in two places, so it's definitely squashed. And on this side, you have to look at it in two different cuts to get it both, but this bone is actually quite separate from the tympanic, so it doesn't have a connection. And we would say it's just accidentally preserved there. And that therefore, where it's sitting is a preservation concern, not necessarily an anatomical concern. Here is the side view of this skull, just to prove that it is really squashed. All the top part of the skull here has been, in the fossilization process, has been squashed down, and we've colored it blue here to be just a series of tiny fragments. So this animal definitely has, after it's been buried, has been altered. So just to review these characters, on this fossil bat, we find that there is not an enlarged cochlea compared to living bats, that it does not have an expanded tip to its stylohyoid bone, and that it does not articulate with the tympanic. So for the three characters that all living echolocating bats have, this bat does not have any of those characters. So we would continue to say that this is not an echolocating bat, and that our flight first hypothesis still holds true, that this was a flying animal but not an echolocator. Okay, so I'm going to do a little bit of the phylogeny thing. Within paleontology, everybody wants to see the characters lined up into a family tree to see how the characters are lined up. So there's programs that can do this for us. We score a whole series of characters, put them into a computer, and it produces a character map for us, which is a branching tree. And if these characters are actually useful characters in terms of understanding the evolution of the group, then the tree that is produced will also be the tree of family relations. So we can do these morphology analyses, but we can also use molecules to do this. So for do this analysis, we've done both, morphology and also molecules. Now obviously there's no molecules for our fossil bats, but there are for the living bats, and that helps us tweak the tree of relationships of these animals. Now, when you do these analyses, you need to compare them to something else to see how do these features line up compared to something that we know is not in the group of these things. So we used a number of different animals, including terrestrial animals like the carnivores and the ungulates and hedgehogs and tree shrews, and these things which are gliding mammals called, unfortunately they're called flying lemurs, but they're actually gliding animals from the Philippines. So we used all the morphology of these things as our outgroup to make a tree of relationships. This is what the tree looks like. You don't need to really see the details, other than these are all the living, previously known bats. These ones with the little crosses are the fossil bats, except for the fruit bats here. And then here is where the new fossil bat lies. So it lies right at the base of the family tree. In other words, no surprise, it's the most primitive bat known to date. When you add the molecules, it tweaks it a little bit better. So morphology and molecules sometimes tell us a slightly different story, but the overall form of this story is really very good. It's very similar. And still, the Onychonicterus falls at the base of the tree. And what that shows us is that since all of these things fly, that evolution of flight happened about here. So all of these people fly. That's the way this tree is structured. And from here on, all of these things can echolocate, with the exception of fruit bats, which I'll get to in a minute. So that shows us this idea that the bats learn to fly first and that they learn to echolocate second. So what does that mean? What is this flying but not echolocating animal? We would say that it may well have evolved gliding first and then flutter gliding and then finally flying, and that it's still probably an insect eater. The study of the teeth of these animals has not been done yet, but it still looks like pretty good standard insectivore-looking teeth. And so we would propose that it would be flying between trees looking for food, and it probably used its claws to climb on trees and glean insects off the bark. Or it may have done this thing called perch hunting, which does not mean it was fishing. It means that it was sitting on a perch and then flying out and catching insects and returning to its perch. So there's a number of living bats that use that approach to catch things. If that's true, if either of these are true, then it has to be using its eyes for finding its prey, which means that it's an animal that's diurnal or living during the day and not living at night. So it's using its eyes, maybe at dawn and dusk, but it's during the day. It's using light and its vision to find the insects. It's either climbing on bark to find them or maybe jumping out and catching them. And we could additionally say that those claws perhaps were used to roost. So since it has claws on all its digits, it may be hanging by its forewings rather than its feet, which is what living bats do. So this is a different kind of animal than any other bat that we've known to date. And these are sort of our speculations. I just want to return to this tree to say I'm going to focus in this region here. And here's an interesting thing. Here's where flight came, and here's where echolocation evolved in the family, except for the fruit bats. So if this tree of relations is true, then we have to propose that the fruit bats actually lost the ability to echolocate, because all its ancestors had the ability to do that, and they do not. So this is an interesting proposal, and it's a byproduct of doing this analysis. So why would fruit bats lose the ability to echolocate? So we're entering the realm of speculation. So far, all fossil fruit bats look like fossil fruit bats. It's the same problem. We don't have any transitional forms for these things. But in terms of the ability to echolocate, physiologically, it's very expensive to do that. It involves not only the flying and breathing and sending out this sound. And so if you can find your food without echolocating, you can save a fair amount of energy by doing that. So we could speculate that these animals may have flown to come to, say, flowers on a tree where there's a lot of insects to eat their food originally, but also found that there was fruit there. And after a time, maybe started specializing on the fruit and realizing that they didn't need echolocation to find fruit. They can pick that out with their eyes during the daytime. And because echolocation is fairly expensive physiologically, it restricts the size of living bats today. And that's why bats are mostly small, because that whole linked phenomenon of sending out signals and flying and breathing are all linked together. And if you can discard that, then you can actually increase your body size. And that's exactly what we see. You need a constant source of food, this fruit, if you're going to do that, and hence you need trees that are fruiting all during the year, which means tropics only. And all of this is what we see in fruit bats today. They only live in the tropics, they eat only fruit, and they have a large body size. So we can speculate that maybe this is what's going on, which is actually very interesting, but we so far have no fossil record to ascertain this. So this is so far a guess. That's all I was going to say today about bats. I just want to acknowledge a few things. One is the Louise Holly Stone Charitable Trust. This is the trust that allowed us to purchase the fossil bat for the ROM. The Madeline Fritz Fund at the ROM was the one that allowed us to travel to Tucson to hobnob with all the dealers. Ian, who did the fantastic preparation. My 3 co-authors, who I haven't mentioned very much, but they are really the bat experts. Nancy is at the American Museum of New York, and she is the acknowledged expert in the world on Eocene bats. Greg Gunnell, who's at the University of Michigan, who also works on basal mammals, and Jörg Habertsetzer, who's done all the scanning in Germany. So the specimens were sent to Germany where he's done the scanning. And finally, I'd like to thank Francois for inviting me here today. Thank you.